I'm going to talk about how ribose and nucleotides are formed and how they come together to create ATP. So, first we have glucose as a starting material, turns into glucose 6-phosphate. Um, two NADP pluses are consumed and turn into two NADPH plus carbon dioxide. Then we get ribulose 5-phosphate, which transformed into ribose 5-phosphate. That's the important intermediate here that enters into a PRPP molecule. The ribose 5-phosphate is an important precursor for nucleotide synthesis, so we'll see how that connects. So this PRPP actually connects over here. The pentose phosphate pathway is important in creation of PRPP, and so we see that product over here. So the PRPP goes in and creates phosphoribosyl amine. In these few reactions, ATP is consumed, and I believe that would come from aerobic respiration or another place that creates some amount of ATP, like the electron transport chain. So with that ATP available, that's consumed. We have two molecules of ATP here that are consumed to create a formylglycinamidine ribonucleotide. In the next step, another ATP is consumed, again coming from electron transport chain, or a place where there's a higher yield of ATP. Creates a 5-amino-imidazole ribonucleotide. Another ATP is consumed to ADP. Various steps are taken. Again, ATP is consumed, and we proceed to create inositol. So inositol is an important precursor for ATP production. So just in terms of the ATP production, we have inositol that takes this pathway with aspartate GTP, which is reduced to GDP plus PI, to adenyl succinate. The adenyl succinase enzyme acts upon this molecule, and with a kinase as well as two ATP that are consumed, an ATP is produced. This is also an important pathway to keep in mind because this is where nucleotide synthesis is occurring. So we've gone through this pathway, but it's important to look at these up here with orotate and carbamoyl phosphate. So those two substances are prerequisites for uridylate production. And so we've seen that uridylate and inositol have been formed through these two pathways. We can split them up into these three pathways. So I should say that where ATP was made, the adenyl succinase was used. Then when we want to make TTP for thymine, thymidylate synthetase is consumed as well as ATP and ADP to create TTP. Then we have kinase. Again, this enzyme is going to utilize ATP for energy, or it's going to catalyze this reaction, as well as using another ATP molecule to create UTP for uracil. And then with CTP synthetase, yet another ATP is going to be used. We can see the importance of ATP in all of these reactions to create CTP for cytidine, or when it is a nucleotide. We can't forget guanine, so the inositol, this other pathway, it uses IMP dehydrogenase, and this time ATP is not used in the synthesis of GTP, at least not in the beginning, I should clarify. So NAD plus water yields NADH plus H plus, and we keep moving with GMP synthetase. Glutamine is utilized as well as ATP to create glutamate and NAD plus PPI, and 2-ATP are reduced to ADP to create GTP for guanine. So we've talked about how ribose and nucleotides are synthesized. So when they come together and through processes like transcription, new DNA can be made, and obviously that's very important for the proliferation of the species. Anyway, so we can come over to other sources of ATP, um, which are glycolysis. So glycolysis is a place where there's glucose metabolism happening as well as gluconeogenesis. Um, so this glucose turns into glucose 6-phosphate. Again, here we have ATP being utilized. It's going to be used twice, and actually 2 molecules of ATP are going to be formed in the creation of pyruvate, which the pyruvate is going to enter into the citric acid cycle or the Krebs cycle to create acetyl-CoA, which will be used actually in amino acid synthesis as well as in an amino acid synthesis. So we see that here, but as well as in the pyruvate dehydrogenase complex. That's what I wanted to say. So in this particular pathway, we have acetyl-CoA, we have NADH being formed, as well as carbon dioxide and FADH2. This is important in ATP production because that's going to enter into the electron transport chain, where the most where when we have aerobic conditions, oxidative phosphorylation will occur, and we'll have the most ATP created, and that's through the action of ATP synthase, which is this structure right here. So the movement of hydrogen ions yields ATP actually. So that's a great and efficient system for ATP production. So I believe that I've hit most of, or if not all of the important places where ATP is formed and how the ribose actually in this creation yields fructose 6-phosphate and GAP. That's going to actually go into glucose. So that's one way we're coming together, or into glycolysis and gluconeogenesis. So that's one way ribose comes together to create ATP. We also see that there is important substances made in amino acid synthesis that will enter back into the aerobic respiration process in the Krebs cycle, and that overall these processes are connected and work together to create ATP and other important proteins for the sustainment of life. Thank you.